Very good morning, students. Today we are going to discuss a topic under Unit 4 in Animal Biotechnology course. And the topic that we are going to discuss is going to be probiotics. So this probiotics is a very well known beneficial factor for humans. And the same probiotics is also going to be a beneficial factors for animals also. So if you look at the term biotics means life. Probiotics is a Greek word. It means it is going to give life. That means it is going to extend the life expectancy, and it is also going to offer the lot of beneficial effects to the animals. And the probiotics are usually commensal bacteria or even microorganisms, which is going to offer only advantage for the host. Why should we use probiotics? The reason for using probiotics is that the number of microorganisms in the probiotics culture, if it is going to be very high, then it can able to compete, it can able to undergo proliferation, outnumber the pathogens that are going to be present in the digestive system. And that is the sole reason for using probiotics. Basically, it directly competes with the uh, interference with the pathogen. So thereby it will eliminate the pathogen, and thereby it offers a beneficial protection. So the probiotics preparations which is used for animal biotechnology is already available in the market, and it is generally composed of microorganisms, especially lactobacilli, and or in combination with streptococci species. In certain cases, it may also contain bifidobacterium and yeast culture. The yeast culture is predominantly used in the cattle industries, and each types of industry with respect to animal biotechnology uses a specific type of the microorganisms. How these probiotics are being used, circulated, it is available in a viable form, freeze-dried form, or in terms of microencapsulated, which will undergo proliferation as soon as it reaches the digestive system of the animal. So, in what way the probiotics is going to be helpful for the host? And that is the sole purpose of using this probiotics. First and foremost important function of this probiotic is it is going to directly act as an antagonist to the pathogen. That means if the commensal microbes numbers are more, in that case, the harmful or pathogenic organism cannot able to attach to the digestive system of the animal. So that is a first and important way by which these probiotics will work. Second, in presence of these probiotics, the metabolism, especially the microbial metabolism in the gastrointestinal tract, is completely changed. That means the beneficial bacteria will produce a metabolite, and this metabolite is going to be inhibitory factor for the pathogen. The third is it also induces or enhance the immune function or enhance the immunity of the animal. And the fourth is it can also directly neutralizes endotoxins that are formed by the pathogenic organism. We'll be discussing this in detail. And apart from these four important ways by which the probiotics work, it can also indirectly increases the growth rate of the animal, and it also increases the feed efficiency, and it also prevents the intestinal infection. So when the animal is undergoing an infection, intestinal infection, whatever the feed that the animal is taking is going to be useless because it is going to be excreted out of the system by undigested form, so that the nutrient uptake is going to be inhibited. So in that way, indirectly, probiotics helps in uptake of the nutrient by the animal. The use of bacterial culture is nowadays increasing very popularly in the poultry industries, wherein the pigs were fed with the microorganisms, and the yeast is also been used in the cattle industry. See, the normal bacterial flora in the animal tract of the pig and birds are going to be found with lactobacilli, streptococci, E. coli, and Clostridium. So, what are the types of microorganisms that are used as probiotics? We can classify that into three types. One, lactic acid producing bacteria, bifidobacteria, and yeast and bacilli. And these common probiotics is going to act as a growth inducing factors. That means in presence of these microorganisms, the growth is going to be increased. And it is also going to act as a protective and synergistic uh, function. The probiotic lactobacilli and bifidobacterium are weakly proteolytic and hence grow relatively slow in milk. The growth of bifidobacterium can be improved by the presence of a suitable companion culture, which in turn can able to secrete proteolytic enzyme, which in turn induces protein hydrolysis, and thereby it increases the production of this growth factor for this microorganisms. And usually the growth substance are going to be carbon, nitrogen source, and growth factor. In certain studies have proved that use of antioxidants improves the efficiency of the probiotics, thereby which in turn increases the overall growth of the animal. So few research have also confirmed that. So the probiotics are used in poultry industry. It is also used in pig. It is also used in cattle, and usually in poultry farming, wherein the chickens are being grown only for the purpose of meat and egg. So it means that meat means it requires more protein. So in presence of there are studies which are shown that in presence of strains like E. coli, which is also going to be non-lethal form of E. coli, had increased the rate of protein synthesis in the poultry industry. So farming the chickens which requires high yield of protein from soya bean. Along with this, if we can able to give the non-pathogenic form of E. coli, it is shown that it increases the rate of protein synthesis for these chickens. So thereby it directly benefits the human who is going to consume the chickens. Okay. Now, what should be the ideal characteristic of a probiotic? That means first and foremost is it should be non-pathogenic to animal, not only to animal and also for humans who used to consume these products. So it should not be a pathogen. Second important property is it should be a gram positive. Why should it should be a gram positive? Because it has a thick membrane so that it can able to resist the destruction of digestive enzymes, which is going to be consumed by the animal. It should have high tolerance for bile, and it should also have high tolerance for acidic pH. It should be also capable of producing lactic acid, and it should be easily undergoing proliferation in vivo condition. That means in the animal it should undergo proliferation, and also in the in vitro condition where when it is mixed with the feed, it should also undergo proliferation. So this proliferation plays a very important role because the more the number of probiotics, it can able to inhibit the growth of a pathogen. I will be discussing that little later in detail how this mechanism works. And it should also have high survival rate after passing through manufacturing process, whether it is in the capsule form or whether it is in the freeze dried form. So it should be surviving in the manufacturing process. And the last one, it should also have high stability at the room temperature, because most of the feeds are going to be kept at room temperature. So when it is mixed, it should not undergo degradation, or it should not have lost its viability. So these are the ideal characteristics of a probiotics. Now, how does the probiotics work? So I told you, if you remember, there are four ways by which the probiotics will work, right? That, but in physiologically, when the organism is under stress condition, what are the stress condition? Parchation is one of the kind of stress these animals used to experience. Castration, weaning, and change in the diet. So these stress and change in the diet may increases the gut pH, which in turn is going to favor the pathogenic organism. So when we have this probiotics, it actually decreases the pH, increases the pH. So thereby the acidic pH is going to be converted into slightly basic pH, which in turn is going to be inhibitory factor for the pathogenic bacteria. Right. So this is one kind of a mechanism of action of probiotics, and the second one is to improve the immune system and neutralizes the toxin. So I'll be discussing this in detail in the forthcoming slide. And another important function is it can able to alter the microbial metabolism, which is very important. So these are the listed potential mechanism of action of probiotics. First, it inhibits the growth of enteric bacteria. How does it inhibits? It decreases the luminal pH, and it also secretes a protein which is going to be cidal for the bacteria, bactericidal. That means if these proteins will kill or will resist the growth of the pathogenic bacteria. It also produces a peptide called as defensin, okay, which in turn is going to fight against the pathogenic bacteria, and it also occupies the space for the pathogenic bacteria. So that's why I told you the number of probiotics are more, then it can able to outnumber the pathogenic bacteria by not giving space for the pathogenic bacteria to attach to the epithelial cells. It blocks the epithelial attachment, and it can also increases the production of a protein called as mucin, which in turn is going to prevent the blockage of binding of this pathogen to the epithelial cells. And it also induces the production of mucus. Mucus is a secretory compound. So when this mucus is produced, the pathogens is going to be trapped with the mucus, and then it is going to be excreted out. So thereby it can also interfere indirectly with the biofilm formation, which is very important for the pathogens to sustain. So these are the mechanism of actions of these probiotics uh, against the pathogenic enteric bacteria. Now, another important point is how does it enhances the immune function of the animal. Number one, it is going to alter the innate and adaptive immune response. How it is going to alter the innate and adaptive immune response? Under innate case, it induces the production of interleukin 10, TGF beta. So these are an important cytokine which plays an important role in controlling the innate immune system. Under acquired immune system, it stimulates the production of an antibody called as IgA. It's a secretory antibody. So when these antibodies are available in the digestive tract, when the antibodies start to secrete in the digestive tract, these antibodies are going to be against these pathogenic strains of these microorganisms. So this will go and bind to these microorganisms. Thereby, it does not allow the microorganisms to attach to the epithelial cells. That is the important way by which it controls or moderates innate and acquired immune response. Apart from that, it also controls inflammatory cytokines like tumor necrosis factor and interferon gamma and so on. It also is known to activate the T cell, okay, which is a part of acquired immune system, and these cells are going to be activated in presence of the probiotics. It is also known to be documented to activate mucosal associated lymphoid tissue. That means kind of an innate immune system, wherein in presence of mucus, the innate immune system is going to be fighting against this pathogen. So these are the ways by which these microorganisms are going to be fighting against the pathogenic strains of the microorganisms, and the mechanisms are shown. And this is the pictorial representation of how this probiotics is going to be working beneficial to the animal. One, colonization resistant. That means it does not allow the pathogen to bind to the epithelial surface. As you can see here, these pathogens are going to be inhibited by binding to this epithelial surface. So thereby, because it is going to act like competitive exclusion, that means more probiotics are going to be there. So it is going to inhibit the binding of this pathogenic strains to the epithelial cells. That is one. Number two, it also maintains a barrier function. As you can see, between two epithelial cells, you have a junction, and we call this as tight junction. So this, in presence of probiotics, it induces the tight junction formation. In presence of claudin, cell addition molecule, it induces the tight junction. So thereby the pathogens cannot enter through the epithelial cells through intracellular. And the third one, I told you, it alters innate and adaptive immune function. In innate, it increases the cytokine productions of IL-10 and TGF beta. And in case of adaptive immunomodulation, it induces the increased production of antibody called as IgA. Not only IgA, it also induces IgM and IgG also. Simultaneously, it also increases the mucin production, which is going to retard the pathogenic bacteria from binding to the epithelial cells. And the fourth important point that I told you is it can able to modulate the signal transduction. So NF-kappa B, mitogen activated protein kinase are going to be activated in presence of the probiotics. And it can also alter the microbial metabolites. So that by how it modulates the microbial metabolites by changing the pH environment of the gut. Okay, and it can also control the quorum sensing mechanisms through which the pathogenic microorganisms can able to sustain. So these are all the overall functions of the probiotics, and we had discussed about the effect of these probiotics, the types of probiotics, and the mechanism of probiotics in this class. I hope this information with respect to the probiotics is going to be highly useful for the students. Thank you, students.